This is page 53, and we've got a diagram of a muscle myofibril. Name the protein in W and X. This is fairly straightforward. You know the names of the thick and the thin proteins. W is myosin, X is actin. And also on this diagram we can just see that Y there, where they're cutting through, is just through this actin. So when we look over the page, we can see where they've cut it through is here in figure 2. So it's just through those actin. And figure 3 shows when the myofibril was cut at the same distance from the Z line at a different stage of contraction. So we notice there's the thin ones and the thick ones. Explain why the pattern of protein filaments differs in figure 2 and 3. So in figure 2 we've just gone through these actins, but in figure 3 it's contracted and these actins and myosins have more overlapped with each other. So in figure 3 there's more overlapping of actin and myosin. The sarcomere, oh I can spell it, sarcomere has got smaller. So you can now see both actin and myosin. And in part C, I'm not quite sure how much you need to know of this, but here we go. Role of calcium ions. So the calcium ions are released from the sarcoplasmic reticulum, which are like the kind of membranes inside around the myofibrils, and then that causes changes to the proteins. So we'll start off with that. So calcium ions released from the sarcoplasmic reticulum. They actually bind to one of our more complicated proteins, troponin, which then causes troponin to change shape, which changes shape, and then that causes another protein, tropomyosin. So this causes the other complicated protein, tropomyosin, to change position. Or change shape. And that exposes the myosin binding sites on the actin. So this exposes the myosin binding sites on the actin, and then they can form their cross bridges. So we can say myosin heads now form cross bridges, and then that will enable the contraction. So I'm not quite sure how much you need to know the names of these proteins, but maybe not quite sure whether you need to know the role of calcium ions.